Established in 1987, the Michael Smith Laboratories, located in the University of British Columbia, is Canada's first interdisciplinary biotechnology unit. One area of research at Michael Smith Labs is lipid nanoparticle technology used in mRNA delivery. Dr. Anna Blakeney is a researcher involved in this field, with whom we were honored to interview and gain insight with her research of the optimization of lipid nanoparticles for saRNA expression and cellular activation using a design of experiment approach. Essentially, her research focuses on the delivery of a small package of viral mRNA, which are strands of genetic material designed to encode for different immune responses. Dr. Blakeney's work uses different lipids, which are molecules of fat, such as phospholipids, ionizable lipids, cholesterol, and pegylated lipids, to determine effective delivery of the mRNA package. The mRNA used in research is known as saRNA, or self-amplifying RNA, which is a more concentrated version of the typical mRNA. Such technology was crucial for the effective vaccination of millions around the world during the COVID-19 pandemic, as the subsequent vaccine developed used this exact mRNA nanoparticle technology.
I'm an assistant professor here at UBC. My lab is in the Michael Smith Labs, and I am also in the School of Biomedical Engineering. And my lab aims to develop next generation RNA vaccines and therapies. So we've already seen RNA vaccines work quite well in the context of the pandemic, but I think there's a lot of ways that RNA vaccines can be improved, and then a lot of other applications that RNA can be used for. So that's what we're exploring with our research.
First, we wanted to ask Dr. Blakeney if research was a field she always wanted to be in.
Yes and no. So RNA vaccines and therapies is still kind of like a relatively new field. And so I was always interested in kind of drug delivery and biomaterials and immunology, as well as preventative medicines. I think that's a really important part of healthcare. And so that's what I've always been interested in. So that kind of ended up in, that's how I ended up in RNA vaccines, but it wasn't like a straight trajectory to there.
Dr. Blakeney then highlights her role in the research paper regarding the optimization of lipid nanoparticle technology.
So my role in the research paper as like the principal investigator or PI on the paper is really to help think about, like, conceptualize the project, and then think about experimental design and how to analyze the data and how to do any troubleshooting that may come along. And so I think the main challenge as a PI is really that you're usually not the one in there doing the research, right? So even though I have a lot of experience at doing this and like could be doing it myself, like part of the goal of doing research and working at a university is training other people how to do it. And so you have to be really effective in the way that you communicate and train people to do these experiments. So I think that's the hardest part, is like, you know how to do it, but you have to like train someone else to do it in the same way that you would. Yeah.
She also highlights the significance of the research and the purpose of lipid nanoparticles in vaccines.
So one of the things we're looking to do with this study specifically is really think about, so we have these lipid nanoparticles that are used in the vaccines, and they've been shown to work really well for RNA vaccines, but we're actually still learning a lot about these lipid nanoparticles and how your immune system recognizes the different lipids that are part of the particles, and then how kind of downstream this affects your immune system. So one of the things we were trying to do in this paper is actually just control kind of how much immune activation you see from these lipid nanoparticles just by changing the composition or the ratios of the different lipids inside the particles. So there's 4 different lipids that people typically use, which is an ionizable lipid, a phospholipid, cholesterol, and a pegylated lipid. And they each have different functions within the particle, so how they preserve the particle stability and encapsulate the RNA. But we're still learning about how your body senses them. So that was what we were really trying to do here, is just get a better understanding of how, when we change these compositions, how does this lead to a different immune response?
We wanted to go a bit more in depth into the research conducted. Thus, we asked Dr. Blakeney regarding the focus on saRNA used and its purpose in the research.
Great question. So, self-amplifying RNA is really just a type of messenger RNA. It's a really long messenger RNA. So, mRNA is usually around like 1 to 2,000 bases, whereas saRNA is huge. It's around 10,000. So, it's much larger, and because of that, it's like a much more delicate molecule as well. But it has some really great advantages. So, because saRNA, the main advantage of it is that once it gets into a cell, it's able to make copies of itself. So this means that if you deliver like one copy of saRNA into a cell, all of a sudden, a few hours later, you actually have thousands of copies of that RNA. So this means this initial dose that you deliver to start off with is much lower than you would need for messenger RNA. It's usually around 100 times lower. So this is the main advantage in the context of vaccines, because as you can imagine, if you were able to get 100 times lower dose, you'd have less side effects due to the RNA. Also from the same size batch of RNA, you'd be able to scale this up and make it go 100 times farther than we currently are able to. So we think this is a really impactful technology.
We were then interested in if there were other types of RNA being developed.
There's just been kind of an explosion of momentum in the field of RNA since the pandemic and the vaccines, because the technology has really been proven now, right? That it can work and that it can be scaled up and safe for billions of people to take. So another type of RNA that people have looked into is called circular RNA. So normally the RNA we use is linear, so it's just there's a start and end to it. Circular RNA is different because it's actually like a circular piece of RNA, as the name implies. And the advantage there is that RNA molecules are sensed and degraded a lot of times by the end of the molecule. So if there is no end, there's no way for some of these mechanisms to detect the RNA. It's still detected in some ways, but it should be much more stable and detected by cells in a different way. So that's another type of RNA that's starting to come online.
To conclude, we wanted to understand the scope of lipid nanoparticle technology research at UBC as well as globally.
Yeah, I think so. What's really cool about being at UBC is there's just a long history of lipid nanoparticles here, right? So Peter Cullis is I would say the godfather of this technology, with obviously lots of contributions from lots of other people, but he's really just built up this technology and started a bunch of companies that do this now. So Vancouver is really a global hub for this. But yeah, you are starting to see many more people working on lipid nanoparticles all over the world now.